Chapter 16 Gene Expression Figure 16.1 The genetic content of each somatic cell in an organism is the same, but not all genes are expressed in every cell. The control of which genes are expressed dictates whether a cell is, for example, an eye cell or, b, a liver cell. It is the differential gene expression patterns that arise in different cells that give rise to, c, a complete organism. Introduction Each somatic cell in the body generally contains the same DNA. A few exceptions include red blood cells, which contain no DNA in their mature state, and some immune system cells that rearrange their DNA while producing antibodies. In general, however, the genes that determine whether you have green eyes, brown hair, and how fast you metabolize food are the same in the cells in your eyes and your liver, even though these organs function quite differently. If each cell has the same DNA, how is it that cells or organs are different? Why do cells in the eye differ so dramatically from cells in the liver? Whereas each cell shares the same genome and DNA sequence, each cell does not turn on, or express, the same set of genes. Each cell type needs a different set of proteins to perform its function. Therefore, only a small subset of proteins is expressed in a cell. For the proteins to be expressed, the DNA must be transcribed into RNA and the RNA must be translated into protein. In a given cell type, not all genes encoded in the DNA are transcribed into RNA or translated into protein because specific cells in our body have specific functions. Specialized proteins that make up the eye, iris, lens, and cornea are only expressed in the eye, whereas the specialized proteins in the heart, pacemaker cells, heart muscle, and valves are only expressed in the heart. At any given time, only a subset of all of the genes encoded by our DNA are expressed and translated into proteins. The expression of specific genes is a highly regulated process with many levels and stages of control. This complexity ensures the proper expression in the proper cell at the proper time. Chapter Outline 16.1 Regulation of Gene Expression 16.2 Prokaryotic Gene Regulation 16.3 Eukaryotic Epigenetic Gene Regulation 16.4 Eukaryotic Transcription Gene Regulation 16.5 Eukaryotic Post-Transcriptional Gene Regulation 16.6 Eukaryotic Translational and Post-Translational Gene Regulation 16.7 Cancer and Gene Regulation 16.1 Regulation of Gene Expression. By the end of this section, you will be able to do the following. Discuss why every cell does not express all of its genes all of the time. Describe how prokaryotic gene regulation occurs at the transcriptional level. Discuss how eukaryotic gene regulation occurs at the epigenetic, transcriptional, post-transcriptional, translational, and post-translational levels. For a cell to function properly, necessary proteins must be synthesized at the proper time and place. All cells control or regulate the synthesis of proteins from information encoded in their DNA. The process of turning on a gene to produce RNA and protein is called gene expression. Whether in a simple unicellular organism or a complex multicellular organism, each cell controls when and how its genes are expressed. For this to occur, there must be internal chemical mechanisms that control when a gene is expressed to make RNA and protein, how much of the protein is made, and when it is time to stop making that protein because it is no longer needed. The regulation of gene expression conserves energy and space. It would require a significant amount of energy for an organism to express every gene at all times, so it is more energy efficient to turn on the genes only when they are required. In addition, only expressing a subset of genes in each cell saves space because DNA must be unwound from its tightly coiled structure to transcribe and translate the DNA. Cells would have to be enormous if every protein were expressed in every cell all the time. The control of gene expression is extremely complex. Malfunctions in this process are detrimental to the cell and can lead to the development of many diseases, including cancer. Prokaryotic versus eukaryotic gene expression. To understand how gene expression is regulated, we must first understand how a gene codes for a functional protein in a cell. The process occurs in both prokaryotic and eukaryotic cells, just in slightly different manners. Prokaryotic organisms are single-celled organisms that lack a cell nucleus, and their DNA therefore floats freely in the cell cytoplasm. To synthesize a protein, the processes of transcription and translation occur almost simultaneously. When the resulting protein is no longer needed, transcription stops. As a result, the primary method to control what type of protein and how much of each protein is expressed in a prokaryotic cell is the regulation of DNA transcription. All of the subsequent steps occur automatically. When more protein is required, more transcription occurs. Therefore, in prokaryotic cells, the control of gene expression is mostly at the transcriptional level. Eukaryotic cells, in contrast, have intracellular organelles that add to their complexity. In eukaryotic cells, the DNA is contained inside the cell's nucleus and there it is transcribed into RNA. The newly synthesized RNA is then transported out of the nucleus into the cytoplasm, where ribosomes translate the RNA into protein. The processes of transcription and translation are physically separated by the nuclear membrane. Transcription occurs only within the nucleus, and translation occurs only outside the nucleus in the cytoplasm. The regulation of gene expression can occur at all stages of the process, figure 16.2. Regulation may occur when the DNA is uncoiled and loosened from nucleosomes to bind transcription factors, epigenetic level. When the RNA is transcribed, transcriptional level. When the RNA is processed and exported to the cytoplasm after it is transcribed, post-transcriptional level. When the RNA is translated into protein, translational level. Or after the protein has been made, post-translational level. 396 Chapter 16, Gene Expression Access for Free at OpenStax.org. Figure 16.2 Regulation in Prokaryotes and Eukaryotes. Prokaryotic transcription and translation occur simultaneously in the cytoplasm, and regulation occurs at the transcriptional level. Eukaryotic gene expression is regulated during transcription and RNA processing, which take place in the nucleus, and during protein translation, which takes place in the cytoplasm. Further regulation may occur through post-translational modifications of proteins. The differences in the regulation of gene expression between prokaryotes and eukaryotes are summarized in Table 16.1. The regulation of gene expression is discussed in detail in subsequent modules. Differences in the regulation of gene expression of prokaryotic and eukaryotic organisms. Eukaryotic organisms lack a membrane-bound nucleus. Contain nucleus. DNA is found in the cytoplasm. DNA is confined to the nuclear compartment. RNA transcription and protein formation occur almost simultaneously. RNA transcription occurs prior to protein formation, and it takes place in the nucleus. Translation of RNA to protein occurs in the cytoplasm. Gene expression is regulated primarily at the transcriptional level. Gene expression is regulated at many levels: epigenetic, transcriptional, nuclear shuttling, post-transcriptional, translational, and post-translational. Table 16.1 Evolution Connection. Evolution of gene regulation. Prokaryotic cells can only regulate gene expression by controlling the amount of transcription. As eukaryotic cells evolved, the complexity of the control of gene expression increased. For example, with the evolution of eukaryotic cells came compartmentalization of important cellular components and cellular processes. A nuclear region that contains the DNA was formed. Transcription and translation were physically separated into two different cellular compartments. It therefore became possible to control gene expression by regulating transcription in the nucleus, and also by controlling the RNA levels and protein translation present outside the nucleus. Most gene regulation is done to conserve cell resources. However, other regulatory processes may be defensive. Cellular processes such as developed to protect the cell from viral or parasitic infections. If the cell could quickly shut off gene expression for a short period of time, it would be able to survive an infection when other organisms could not. Therefore, the organism 16.1, Regulation of Gene Expression 397 evolved a new process that helped it survive, and it was able to pass this new development to offspring.